Hello, this is Arkasha from MCAT Simplified. Today we're going to be discussing the isoelectric point and then also amino acid titrations. So to understand isoelectric points, we have to first introduce this concept of a zwitterion. So a zwitterion here is basically when you have a molecule that has both positive and negative charges, usually one positive and one negative charge, but the overall charge of the molecule is neutral, 0, because they all cancel out, all right? And amino acids are great examples of molecules that can form zwitterions, all right? Now, the isoelectric point, Pi, okay, corresponds to the pH at which the molecule that can form a zwitterion is predominantly found in that neutral zwitterion form. So it's the pH at which, in this case, the amino acid or the peptide is going to predominate in its zwitterion form, all right? So to illustrate this, let's look at glycine, the simplest amino acid, all right? So glycine, when, for example, the pH is equal to the physiological pH of 7.4, notice that at that point, its amino group is protonated, positive charged, and then its carboxyl group is deprotonated, negatively charged. And of course, if it has a positive charge and a negative charge, those two cancel out and you get a neutral charge overall. So there, glycine is existing as a zwitterion, all right? Now, notice, right, that you have two pKa's. pKa1 corresponding to the pKa of the carboxyl group, because remember, that oxygen can be protonated or deprotonated. And we're going to say that that equals approximately 2. And then the amino group pKa, which is pKa2, again, because that nitrogen can be protonated or deprotonated, it has a pKa of 10, approximately. Now, these are estimates. In real life, it's established what the pKa values are for these different groups, for the amino group, the carboxyl group, and then if you have side chains that are ionizable, those also have pKa's. And there's charts for that. They're experimentally determined. Typically, you might be given that information if you're asked a question about this on a test or on the MCAT, okay? But for the sake of example, we'll use these nice numbers. And this is approximately what the real pKa's would be for the backbone amino and carboxyl group in any case, okay? Now, some really golden rules to remember about situations with zwitterions is that if the pH, okay, is less than the pKa of the group is going to exist predominantly in a deprotonated form, all right? Whereas if the pH is greater than the pKa, then the group is going to be primarily deprotonated, all right? So pH less than pKa, group is going to be protonated predominantly. pH greater than the pKa, the group is going to be predominantly deprotonated. And then, of course, if the pH is equal to the pKa, exactly half of the group is going to be protonated, or exactly half of the molecules will have a protonated form of that group, and half of the molecules are going to have a deprotonated form of that group, all right? Now, talking about amino acid titration, right? So with any titration, we're going to imagine that we have the molecule in a solution, in this case, an amino acid, and that solution exists at, a pH of some sort. In this case, we're going to start at 0, all right? And then we're going to add known amounts of, in this case, base or a titrant, okay? And we're going to see how the pH changes, all right? And this is going to be important for experimentally determining the isoelectric points, all right? And we'll be able to see using this titration curve where the isoelectric point for glycine, and really this will be true for all neutral amino acids, okay, without ionizable side chains, where the Pi is going to be, all right, the isoelectric point. So, and this is really a good way to approach any problems where you are asked to find the isoelectric point, be it of an amino acid or even a whole peptide, okay? It's to pretend that you're starting at a low pH, an acidic pH of like 0, where it is lower than all of the pKa's of every group present, okay? Because then you know that, again, applying these rules, that if the pH is lower than any pKa, that group is going to be protonated, all right? And then what you'll do is you'll increase the pH over and over again to see how, when at what pHs the groups are going to be protonated and which ones are going to be deprotonated, and calculate the net charge at each of those pHs. And we're going to apply that process with glycine. So at a pH of 0, corresponding to here, that pH is going to be lower than the pKa of this carboxyl group, right, which we're saying is around 2. And then it's going to be lower than the pKa of the amino group, of course, which we'll say is 10. And so both are protonated. And when this is protonated, it's neutral. But then when this is protonated, it's positively charged. So the net charge here is +1. All right. Now, notice that on the titration curve, as we add base, right, we're adding more concentration of base going that way. And as we do that, initially the pH is increasing pretty rapidly, but then we get to this zone here where there's really not significant changes of pH anymore. And that's because we're entering a buffer region where the base is reacting with this group here, which has the lowest pKa, okay? And it is deprotonating it, okay? So you're going to get some conjugate base. And that's going to create a buffer solution, all right? And here, notice that when you have exactly equal amounts of the deprotonated form of this and the protonated form of this, the pH is actually going to be equal to the pKa1 of this group, okay? So as we can see, since we're saying the pKa1 is 2, that's where the middle point of this buffer region is, all right? But then eventually, the base will, if we add enough base, it will react with all of this carboxyl group, and all of this glycine will be converted into the deprotonated form here. And then the pH is going to start to rapidly increase if you continue to add base. And it'll continue to rapidly increase until eventually, right, you're going to get to when this group, the amino group, starts to be deprotonated, okay? But before we get to that, notice that if we increase, okay, if we increase the pH above the pKa of this carboxyl group, and we can say, since there's a big, if the pKa is 2, we can increase it to 3, to 4, to 5, whatever, to 6. Then notice that this group starts to get deprotonated. All right, that's what we talked about with this buffer region here. It's going to start to get deprotonated, and you're going to have species like this where you're going to have some deprotonated form of the carboxyl group. And then, of course, the amino group is still going to be protonated because its pKa is like 10. So the pH is still lower. All right. And then you're going to get this zwitterion form where it's neutral. All right. But then again, after eventually when you increase the pH high enough where the pH is now going to be greater than the pKa2 of the amino group, so let's say the pH is 12, in that case, this amino group will now be deprotonated too. But then, of course, this carboxyl group is still going to be deprotonated, so it's negatively charged. But now we've lost that positive charge, so the overall charge is -1, all right? Now, as it turns out, to calculate the isoelectric point, which remember, that's going to be the pH at which the dominant form in the solution of the species, the molecule, is the zwitterion form, the neutral overall form, which is this form here, right? To calculate that, we simply take pKa1, add to a pKa2, and divide by 2, so you average them, and that's going to be the pi. And as it so happens, for these numbers that we're using, where pKa1 is 2 and pKa1 is 2 and pKa2 is 10, 10 plus 2 is 12, divided by 2 is 6. So for these numbers, which again are not exact numbers, but for this example, the pi of this glycine is going to be 6. Now, another way to consider this is that essentially the neutral form, okay, the pH at which the neutral form predominates is going to be one where it's sort of between 2 points, point A and point B, where point A, okay, is going to be where exactly half of the species in the solution have a +1 charge overall, and exactly half are neutral, okay? And then that's point A. And then point B, okay, is going to be the point where exactly half of the species are neutral, and then half of them have a -1 charge overall. So between point A and point B, exactly halfway between those 2 points, that's going to be where the zwitterion form is going to predominate, exactly halfway between those 2 points. That's another way of explaining that. And really, for any neutral amino acid, be it, like I said, glycine here, or serine, or tyrosine, or, any neutral amino acid, you are able to use this exact same process because you're only worried about ionizable groups in the backbone. The side chains can't be ionized. So for any neutral amino acid, the pi is always going to be equal to the pKa1 of the carboxyl group plus the pKa2 of the amino group divided by 2. However, if you introduce side chains that are ionizable, this process changes, and that's what we'll cover in the next video. And that's our video on the isoelectric point in amino acid titrations. Check out our website at mcatsimplified.com.